Thanks very much. It's a great pleasure to be here. I would like to thank the organizers sincerely for allowing me to present. I do have to tell you that I missed the bulk of this meeting. I was working last night on a grant, an Alzheimer's teen grant. Set my alarm for 5:30. Woke up at 7 with it bright outside, and then looked at my alarm, and it was 5:30 p.m. So quite a rush to get in here this morning. So I apologize if I'm jittery and sweaty and all those things. I would like to just make a little comment about my Canada Research Chair. This is the chair in neurodegeneration and protein misfolding diseases. My lovely wife, Rosie, said, "Oh, that's too long. Why don't we just shorten it to your misfolding chair?" So, I do want to mention disclosures, but I hate the word disclosure because it sounds so furtive. So I'm going to put it right out there that I'm highly industrially engaged. I started 2 Canadian biotech companies, both of which are still alive in this climate. That's no mean achievement. I've licensed technology to Biogen IDEC, also to CanGene Corporation, which is a successful biotech company in Canada. And I serve on the scientific advisory board of Prothena Biosciences. I'm going to ride roughshod over a huge topic, which is protein misfolding diseases, but I'm going to give you a little bit of introduction to where this all started with the transmissible spongiform encephalopathies. This is a group of infectious diseases that produce neurodegeneration, death of nerve cells. And they're very strange because they're not associated with inflammation. So you can, of course, conceive of an encephalitis where you catch a virus, there's this massive inflammation, nerve cells die, it's acute. But the transmissible spongiform encephalopathies have a long incubation period, a relatively protracted disease course. They result in neurodegeneration with this hallmark of spongiform change, which is little sponge-like lesions in the brain. And they're irreversible, untreatable, and unassociated with inflammation. We've known about one of these animal diseases, sheep scrapie. Goodness, how did that happen? There it is, okay. Sheep scrapie for, this is going to be a problem. For about 300 years, this is a disease of sheep and goats that can be transmitted in the laboratory experimentally and also between animals. This results in a neurodegeneration and ultimate death of the animal. As time has passed, as the centuries have passed, there has been a recognition that sheep scrapie is not the only animal prion disease or transmissible spongiform encephalopathy. Of course, you'd have to be living in a box not to know about bovine spongiform encephalopathy, the thing that ground our cattle industry to a halt back in 2003. With an ultimate loss of about $10 billion to the economy. But perhaps you don't know about some of the human disorders. This group started with descriptions of Carleton Gajdusek, who won the first Nobel Prize in this area, of Kuru, a disease of Foray people, a Stone Age linguistic group in Papua New Guinea, who practiced the consumption of deceased relatives as part of their mourning rites. Yep, you heard that correctly. And women and children would wind up eating the brains, which were the least attractive component, and come down with this transmissible spongiform encephalopathy called Kuru. Gajdusek actually recognized that there were features in common with Kuru and Scrapie, and injected non-human primates with Kuru brain homogenates and transmitted the disease. Since that time, Creutzfeldt-Jakob disease, Gerstmann-Streusler syndrome, fatal familial insomnia have been recognized. And finally, in 1996, a human form of bovine spongiform encephalopathy called variant Creutzfeldt-Jakob disease. This disease has claimed about 200 people around the planet and appears to be in its wane at this point, with the recognition that contaminated cattle material was responsible for transmission of this disease. So if this group of diseases is not a virus or not a conventional infectious agent, what could it be? And here we move to the age of Stan Prusner, who won the second Nobel Prize in this area. What Dr. Prusner first hypothesized, and he's the type of guy who publishes his hypothesis papers in Science, back in 1982, he said, the infectious agent appears to co-purify with a protein. Could it be that this group of diseases are due to a transmissible protein? So he spent his professional lifetime working in this area and eventually eked out what we know as the Prion hypothesis, which has been validated so many times it should probably be a law rather than a hypothesis. But in this scheme, there's a normal cellular protein, which we all possess. It's ubiquitously expressed. It's most highly expressed in the brain. This is rich in alpha helical content. The protein itself kind of has a lot of these motifs of bed springs in it, hopefully conveyed by this. This protein can be refolded or misfolded into an abnormal disease-associated form. And in this form, the protein changes into a beta sheet-rich molecule, which becomes highly protease-resistant, highly insoluble, but it's the acquisition of another property that makes it epic setting in biology, and that is the ability to recruit more copies of itself from the normal cellular isoform. So here we have this pictured as a kind of catalytic agent. When the bad guy comes in contact with the good guy is converted into more of the bad guy. And you could see where this would be a kind of, logarithmic or exponential production of infectious material. It's almost like it's replicating, but it's not real biological replication. It's a template-directed conversion which produces more of the infectious form. So I entered this field a long time ago. I've published on the cell biology of the normal cellular isoform of the prion protein back in 1990. But in subsequent times, me and my lab have spent a lot of time trying to map parts of the prion protein that are exposed when it's misfolded in disease and buried in the normal form of the prion protein. And back in 2003, we published, 10 years ago, we published in Nature Medicine the first hypothesis-driven misfolding specific epitope, immunologic epitope of the prion protein, which turned out to be a very small beta strand, just 3 amino acids, which were exposed which were buried in the normal form and exposed when the protein was misfolded in disease. We went on to map the other beta strand as a prion-specific epitope, and more recently, a loop, which is compact, not accessible to antibodies in the normal form of the prion protein, but becomes unstructured, antibody accessible in the misfolding or unfolding that is consequent with prion protein conversion in disease. Why is this important? Well, we figured that if we were able to precisely know what parts of this protein are exposed when it's misfolded in disease, we might be able to immunologically target that and create vaccines and immunotherapies. Here we have, through the work of the VIDO group, Vaccine and Infectious Diseases Organization, and especially Scott Napper, we have a vaccine candidate to prevent scrapie in sheep and, more importantly, chronic wasting disease in deer and elk, which is spreading in the wild like wildfire. This was a good accomplishment of PRION-NET Canada, which was a 7-year network of centers of excellence. I believe that we have a way of preventing these diseases in animals. But subsequently, we and many other laboratories have gone on to consider the idea that the prion hypothesis is not a one-trick pony, that it might apply to other diseases, especially neurodegenerative diseases. And in this generalized scheme, we all know that proteins start out as a string of amino acids. They acquire their function when they're folded into a proper three-dimensional shape. So this loose chain acquires a structure which is responsible for the normal functioning of that protein. This loose chain doesn't have any function. But when a protein is folded into its proper three-dimensional shape, that used to be thought of as the end. That's what happens. But we're starting to understand that in many diseases, there's a template-assisted misfolding of the normally folded protein. And this, there's literature that this applies to Alzheimer's disease, which we'll discuss in more detail, Parkinson's disease, which I won't discuss in great detail, and my favorite disease to hate, amyotrophic lateral sclerosis, and I'll show you some data on that. We've also considered the idea that misfolding or unfolding could be local, it could be dynamic. So here's a protein that unfolds in one little corner and then folds back into the normal form and then unfolds and keeps vacillating between these states. And we're doing work connecting this local unfolding to cancer, and I'll show you a few slides on that. Now, what's the big deal? Why should we be interested in this potential mechanism for neurodegeneration and cancer? Here's the second disruptive idea, that if we can identify these regions which are exposed in the abnormal form, even dynamically in cancer, then we can immunologically target them. And why do I call this disruptive? Because for the last 100 years or so, we've kind of gotten into the pharmaceutical mindset that we're drugging small, well-defined targets, a pit or a cleft or something like that on the surface of a relatively stable protein like an enzyme or a receptor. When we're dealing with misfolded proteins, small molecules just throw up their hands and walk away. Here's a big floppy target, not something that's small and well-defined. And this is what an antibody eats for breakfast. A large floppy target can be recognized by an antibody. So this has some advantages of presumed efficacy because you can neutralize the toxicity of misfolded proteins, block propagation, speed up their degradation, whatnot. It also comes with the advantage of safety, that the antibody is not recognizing the normal form and sparing the cell and the body from massive autoimmunity, preserving the normal function of the protein. So this, as you saw before, is being applied in a prion vaccine, mapping these misfolding specific epitopes of the prion protein. I'll show you a few slides where my company, Amorphix, is applying this idea to cancer, and I'll show you a fair amount of detail on a misfolding specific epitope for A-beta oligomers and misfolding specific epitopes for superoxide dismutase 1. So now we turn to Alzheimer's disease. This is, I don't need to introduce to this audience that this is bad. Alzheimer's disease is costing little tiny Canada about $15 billion a year, which will in a generation be 10 times that. We know that if we figured this out, that if you can delay the onset of Alzheimer's by just 1 year, you could shave $1 billion off of our healthcare provision in Canada and $10 billion in the U.S. So this is an enormous problem, but it comes with enormous opportunity if we were able to effectively treat or prevent Alzheimer's disease. What is the evidence that Alzheimer's actually progresses through a prion-like mechanism? Well, surprisingly, there's a lot of it. This started with Tycho Brahe back in the 80s, who showed stages of Alzheimer's where there would be tau misfolding and phosphorylation and aggregation that starts in the hippocampus and then spreads through neural pathways to engage the entire association cortex of the brain. So this is a process that spreads from region to region, cell to cell. Very similar to prions. We also know experimentally through the work of Larry Walker and Matthias Eucker that a Alzheimer's brain homogenate injected in a mouse model of Alzheimer's is able to produce a spreading amyloidosis. Here shown in a paper from Science in 2006, and shown by Larry Walker as far back as the year 2000. Now this factor can be depleted with antibodies against A-beta, a little fragment of the amyloid precursor protein, and it can be blocked by boiling. So we know something about this seeding factor, that it contains A-beta, and it's a non-covalent assemblage of A-beta. In other words, an aggregate without a chemical bond between the monomers. This work was extended by Claudio Soto more recently to show that animals or mice expressing the human amyloid precursor protein, the wild-type amyloid precursor protein, could also show this spreading amyloidosis when injected with Alzheimer's brain homogenates. So what could be an assemblage of A beta? We know that A beta is a fragment of the amyloid precursor protein. Many, most or all familial Alzheimer's disease is due to defects in the processing of A beta or its clearance. We know that A beta collects in amyloid plaques. This has been known for over 100 years. And here it's in a fibrillar form. But more recently, we understand that there's an unstable intermediate between the monomer A beta and the fibrillar plaque, which are small aggregates, oligomers of A beta. And these small aggregates are able to interact with synapses and cause them toxicity. These small aggregates, oligomers, are also able to kill neurons. It turns out the monomer and the fibril form of A beta are also toxic, but less than 1,000-fold less than oligomers, which are soluble and can travel from region to region in the brain and induce toxicity. So we set it upon ourselves to try to figure out what might be an oligomer-specific epitope, to try to specifically target oligomers to neutralize their toxicity and perhaps neutralize their ability to seed the propagation of Alzheimer's disease. And we've hit up against a problem that has bedeviled structural biologists forever. And this is from the work of David Eisenberg, who's probably the premier structural biologist, X-ray diffraction expert in the world. He wrote an article in JACS just discussing the problems with trying to figure out the structure of A beta oligomers. Major problems are that there isn't one oligomer form, there's hundreds. They rapidly interchange between monomers, other oligomer forms. They're quite dynamic. They're probably at least partly amorphous, so they're not the sort of thing that you can get an atomic level structure on. So he laments the dynamic, polymorphic, non-crystalline behavior. There's no worse behavior for a crystallographer than to be non-crystalline. Of oligomeric species hinder structural studies at atomic resolution. So me and my colleagues went on an extended meditation of what an oligomer-specific epitope might look like. And we studied the known fibrillar structures which are crystalline, or at least paracrystalline, so their structure is well known. The structure of fibrils is two beta strands with a beta turn, which is exposed to solvent. These amino acids are all exposed to solvent, with the exception of a charged lysine at position 28. This charged lysine is engaged in a salt bridge, a kind of ionic interaction with another charged, oppositely charged, positively charged aspartate at position 23. So there's two charges which are buried in the interior of this hydrophobic structure. They're shielded from interaction with solvent, and this makes perfect sense for a fibrillar species, for a species that precipitates out of solution, that just, sticks like a bump on a log in a plaque. But it does not make perfect sense for a relatively unstable, soluble species of which oligomers are. So we reasoned that in oligomers that this turn was exposed, just like it would be in A-beta fibrils, but there would be a disruption of the internal salt bridge with the lysine pointing out into solvent. So we modeled this structure. We actually made a cyclic peptide in which the amino acids at the turn are exposed to solvent and the lysine is also exposed to solvent. This lysine can't possibly go inside and bury itself inside the cycle. It must protrude into solvent. And that's a hypothetical epitope, immunologic epitope, composed again of 3 amino acids, which are structurally constrained. So we raised antibodies against that structurally constrained cyclic trimer, and we found that the antibodies reacted with synthetic oligomers that we made in the lab. They also reacted with authentic oligomers extracted by saline out of Alzheimer's brain, present in Alzheimer's CSF but not control. The antibodies did not bind to fibrils. They also didn't bind to monomers of A-beta. And most importantly, or pretty importantly, they didn't bind to the amyloid precursor protein. So this is a protein which is present at the surface of cells. We wanted to avoid autoimmune consequences of reacting to a cell surface protein. Incredibly, these antibodies against the 3 amino acids were able to block toxicity of A-beta oligomers both in vitro and in vivo. And we have preliminary data that these antibodies block the seeding activity of A-beta oligomers in Alzheimer's mouse models. So this is a remarkable assemblage of data that points to the high likelihood that we have identified an oligomer-specific epitope which could be useful in diagnostics, could be useful in therapeutics, and this package was licensed by CanGene Corporation last year to develop an immunotherapy for Alzheimer's disease. So we've also been working at my company, Amorphix, on an oligomer assay, and we find that aggregated A-beta is present in mild cognitive impairment and Alzheimer's disease, and not present in age-matched normal controls. So this is right from the get-go better than the conventional tests for Alzheimer's disease, which are soluble A-beta 42, tau, and phospho-tau. So now we'll switch gears to amyotrophic lateral sclerosis. And I've directed and served in ALS clinics for 25 years now. So I've held the hands of about 5,000 people who have died of the disease. This is a progressive paralyzing disease. We have no cure. We have a treatment which is called riluzole. It's a small molecule that slows progression of the disease by about 10%. So, in other words, instead of getting worse at 100% your rate, you're only getting worse at 90% your rate. Costs of ALS, because it's paralyzing, because it often affects people at their peak work ability, is astronomical. just the nursing and caring for a patient with ALS can reach $200,000 a year in the United States. Here in Canada, we have a series of wonderful ALS-centric clinics, so this care is provided through our medical care system. Is ALS a prion-like disorder? Well, this kind of entered my consciousness many, many years ago with the very uncanny experience of seeing ALS patients present with weakness in their left big toe or their right index finger, and having the disease creep over the neuroaxis until everything's involved, including the muscles of speech and swallowing and respiration. So I was working in a Prion lab at the time and working in an ALS clinic, and so it struck me that there are many features in common with ALS and the progression of prion diseases. This notion was really systematized by John Ravitz over in the last decade. But using pathological specimens, he was able to show that ALS does indeed progress in a spatially and temporally restricted manner. So this has now entered the literature as something that we all know instead of a one-off clinical observation. So if we're going to consider ALS as a prion-like disease, what do we have as candidates? So unlike Alzheimer's disease, unlike the prion diseases, the familial prion diseases, there is a bewildering array of genes which can be mutant in ALS or even predisposed to ALS. This has been reviewed. So there's an ongoing effort to discover new genes in ALS, but what many of us feel is that the mutations are in three general classes. There's a class for protein processing, trafficking, degradation. There's a class for RNA processing, trafficking, and degradation. And then there's a single gene in the third class, which is superoxide dismutase 1. I think this might be trying to tell us something, that SOD1 is in a class by itself, and one can conceive the other genes as somehow affecting the stability or folding of SOD1. So we went after SOD1. This is many years now, and we tried to identify regions of SOD1 that are exposed when the protein is misfolded in disease. If it sounds familiar, it is familiar. We were able to identify many epitopes which are exposed when SOD1 is misfolded, but I will tell you about two of them, which are at the C-terminus of the protein. Yeah. These have been published in Nature Medicine and PNAS. We also took advantage of a frameshift mutation called G127X that lacks this entire C-terminal region. And I'll just show you briefly that when we expressed G127X in normal human cells, we found that that was associated with the misfolding of wild-type SOD1. We published this in PNAS back in 2011. This protein, misfolded protein, becomes sensitive to protease digestion, just the opposite of the prion protein. And we find misfolded SOD1 in sporadic ALS as well as in familial ALS. So all that to say that we have identified that some of these epitopes can slow disease, to significantly slow disease in mouse models of SOD1, and this is being developed as a passive immunotherapy by Biogen. So I think I'm not going to get to cancer, but I just want to conclude with neurodegeneration, that there's this enormous window which seems to be opening up for us, all based on understanding of prion diseases. So Alzheimer's, Parkinson's, ALS do appear to have a propagation of protein misfolding with a different protein in the starring role for each disease. But one can also make a case for type 2 diabetes, schizophrenia, Huntington's disease, amyloid neuropathy, and whatnot. And I just want to show you very simple-minded what we're proposing, that propagated protein misfolding can start in a cell, travel to another cell, and kill that cell as well as the starting cell. But if we have an antibody or a vaccine which has access to the misfolded protein in the space between cells, we can neutralize that transmission activity and the nerve cells protected. So I think I only have 2 minutes and 30 seconds left. If you would like me to stop here and take questions, I can do that. If you would like to see a little bit of cancer, I can do that too.
What would be the cancer if we see at the end will come to the coffee break for questions?
Okay. Unfortunately, I will not be here at the coffee break. I'm giving another talk in Quebec City in the afternoon. So, do the cancer. Okay. So it is reasonable that cancer cells are subjected to many environmental and intrinsic factors which can misfold proteins. I will mention a few of these, that there's a low pH environment around cancer cells. Proteins are aberrantly glycosylated. Quality control for proteins is defective in cancer cells compared to normal cells. But what we did was to apply our reasoning about looking for misfolding specific epitopes in a computational algorithm, which enabled us to produce energy landscapes here, and this is of the prion protein, but the peaks are extremely stable, the valleys are relatively unstable. So we predicted that if a protein on a cancer cell or anywhere is going to misfold or unfold, it's going to be in these valleys. So we raised antibodies against the valley epitopes, and here is the prion protein again. As I told you, we experimentally discovered the first 2 epitopes, but we used the computational algorithm to discover this 3rd epitope, which is a compact loop. We raised antibodies against this compact loop and showed that they are able to significantly slow the rate of ovarian tumor growth in a mouse model of ovarian cancer. And here is a photomicrograph of the antibody finding the tumor with the brown color, but sparing the normal tissue outside. So we've also done work to show that these epitopes, these misfolding specific epitopes, are partially unfolded at the surface of cancer cells using proteinase K. So I'm afraid that's all there is. This work is is the result of a number of very stimulating, wonderful collaborations, and the funders you can see, CIHR, ALS Canada, PRION-LIKE Canada,